5 minutes to get this prediction in of what is the new species of the week. And I will show you the dreaded clue that was provided today.
That is not in focus. I don't know. What am I supposed to do with that?
There you go.
What are my options?
Your options, Lita. Coral, sponge, squid, crab, beetle. Y'all see this? Look.
Keep my opinion until the end.
So this is the highest res focus that we were able to get, y'all. So this is in the paper. There are some really, really beautiful images because there's a lot of scanning electron microscopy that was done. So that's, remember, y'all, when you dry out the specimen, or if you're using like an insect exoskeleton, it's still dried out, but you don't have to worry about keeping the integrity of the structure. And then you just spray on a little bit of gold, throw it into the electron microscope machine, and it shoots electron beams. There's a sensor that gets you like these really, really high, beautiful resolution images where we can see like a ton of detail. It's really cool. And so this paper was actually in Nature, of all things.
Interesting.
And not only did they do the scanning electron microscope images, but they did like actual images of the organism in the location where they discovered it. So you get to see its native form. Well, coral, sponge, squid, crab, worm, beetle, and fern.
All right. I see that most of the party people selected coral, and that was going to be my guess. So now I feel like I ought to be contrarian, but I not.
Those 80% of the party people suggested coral.
All right. My head is telling me it's not a fern, but my heart is telling me I'm going to say fern.
So let me zoom out, Lita.
All right.
There it is, zoomed out.
Ooh.
And then here is the title of the paper. New carnivorous sponges.
That was what my head was telling me was second choice. Because the background looked aquatic, didn't it? It did look like an aquatic background. And okay, all right, here we go.
Sponge.
Sponge.
And it's a carnivorous sponge, which also is an aside. What? What now? There's carnivorous sponges? Yeah, there are. So this paper, y'all, it was this, if you remember a few weeks ago, maybe a month or two ago, Lita told y'all about a mysterious disappearing jellyfish. Do you remember this, Lita? That they kept bringing the jellyfish up.
Oh, yeah. The deep sea jellyfish. Yes, it would dissolve. They would vanish. They'd capture it in the deep ocean and they'd bring it up in a submersible. And by the time they got to the surface, it was just gone. And it was because it was disintegrating because of it couldn't tolerate its structural integrity, couldn't handle the lower pressure at the sea level.
So as it was being brought up, it came apart.
Yeah.
So what's really cool is it turned out these sponges also had this issue that if you brought them up, they would have like the structural integrity would be ruined. Mm-hmm.
But they don't actually dissolve, I take it.
They don't dissolve. Yeah, carnivorous sponges discovered off the coast of Australia, and they were actually by a remotely operated vehicle, or ROV, and it turned out they wanted to discover them like this, because when they brought up the specimen, they went just kind of, they weren't able to survive at the like lower pressure that we have up here at the surface. They needed the really high pressure near the bottom of the ocean to survive. They didn't dissolve like your jellyfish.
Well, okay, so here's the question. When I was reading up about that jellyfish situation, sometimes with animals, it's the pressure change is too rapid. And so there are sort of like titering ways that you can get around that. Or is it actually like in the case of the jellyfish, the membranes that hold its cells together cannot stay structurally intact at sea level pressure. It's physiologically impossible for them. So there's no amount of coddling and hand holding that you can do to get them up to survive at that level. So did they try anything like that? Was it just because the pressure change itself, if too sudden, can devastate a lot of different creatures?
Yeah, they went slowly, but still wasn't able to survive.
Okay, and I'm assuming they didn't figure out what it was.
It wasn't the point of the paper for this. They did make a really cool effort, though, within the paper. Like if we scroll down, there's this. So this is the map of the different regions that they looked at for the 4 new species. This is a heat map as well. of where they were looking at between the two locations. So they're 1,000 kilometers apart. The range where they found these 4 species.
Wait, I'm confused by the heat. Showing me a heat map for what? Where they were looking? Is it an actual temperature map?
Sorry, heat map in terms of depth.
Okay, so like a topological representation.
Yes, temperature.
Well, because normally a heat map is like concentration or density, and I was like, I don't understand what we're. Okay, we're fine now.
Depthity.
I'm all up to date now, chat.
And then they had a bunch of images here. So these were some of the scanning electron microscope images of elements of the sponges.
Wait, okay. If it didn't survive, how did they get samples for EM?
There's calcified. So they're not looking at like the soft tissue,
okay,
but they're looking at the hard remnants.
Okay.
And so from this, it's not as apparent. So this is just a comparison of the 4 different ones.
Okay.
And showing in some of those regions. But then they have, here we go, these kinds of images. And these I think are really, really cool because they've got an A and B, the actual animal, it taken at an image at the depth at which it's alive. And then they have the bring up, but they're doing even like a light microscope for panel E, and then they zoom in for the scanning electron microscope. So you can see like a really, really fine detail. The sequencing they did for this is the one that you're not a fan of, that they did like the ribosomal subunit and the mitochondrial segment, so that they were different species. They didn't do the whole genome.
I will say though, like I love SEM images. It's super high res though. I guarantee you they could like blow it up to be the size of this green screen and it would still be crystal clear. SEM usually creates some really magnificent.
But then they like zoomed in on some of the calcified bits as well, like the unique properties of them. Like this is like the needle element to it. These are like the little teeth that they use to like capture the prey. And then for each of these 4 species, they had these kind of images. So like I really liked these because I thought it shows you by eye the diversity of what they look like, not just the zoomed in region of interest like with the SEM, but like the whole organism.
So did they come here looking? I'm guessing they were on it like an ROV expedition. They were just identifying new species and they were like, hey, we also found a bunch of new sponges. Let's do a paper on sponges. Is that?
Yeah. So
it wasn't like it's targeted. We're looking for sponges.
This is part of this
oceanographic.
I think you've actually had some of a new species.
Oh yeah, I check out their stuff all the time. They're always doing.
Yeah, and we may have even, y'all, honestly, there was one week where we did like a press release on new species that they found, but it wasn't a published paper yet. This isn't that one, but this is because this is looking at the carnivorous sponges. But are they endangered? That's a great question, Sparky. This one did not go into an attempt at a survey of what numbers they have. We have had papers in the past that have actually dissected like how likely is it if they're endangered. And I remember we found we had one paper of a new species that was like, hey, this is brand new and it's extinct. And then we've also had ones where it's, hey, this is brand new and it's critically endangered. This is more like the standard paper where it's like, hey, it's brand new. We don't know.
Yeah, it's so new that they have no idea as to how many are out there.
It's also hard with these, I feel, because they're at such depths. Like it was only with the rover that they were able to identify the specimens. And so you can imagine how difficult it would be to drive like a little remote control car around on the ocean floor to try to find these specimens. And I mean,
by a remote control car or a remote control submarine driving on the floor.
I guess when I saw ROV, because I've heard that term used for like the Mars. Yeah, so I just assumed it was. So again, I thought, let's see, what was at the specific habitat? Flashy for rocks or smokers? Cliff Alistair McLean, great question. No smokers at any of them. So like those hydrothermal vents were not in the area. It was like this rocky domain. Like it was just and the way they just described it too is like it had a lot of like different kinds of rocks and like the depth that they were showing. But there wasn't anything to suggest there was like the really high temperatures that are sticking out there. Do you know what these eat? They found what was it? It was like other animal remains in them, but it was like a wide range. They weren't quite sure of all of the different flavors out of it.